Greetings, nerdlings. This is going to be my first podcast for you. Some of you seem to be having a little bit of trouble with all of the biomolecules and some of the carbon bonds, so I wanted to go ahead and make another podcast, and I will be walking you through that. So, life substances. Everything that is living is actually made out of carbon. C for carbon. We also have quite a few other elements that help compose living organisms. So anything that has carbon in it is considered to be organic, meaning that it is of life. So we have a carbon skeleton with hydrogen, oxygen, nitrogen, and phosphorus, as well as sulfur that can be attached. And the acronym for that is CHNOPS. That's one of the ways that you can remember all of the elements that help compose living organisms. So all of these, like I said, are found in living things. And then organic chemistry is the study of all living things. So the structure of carbon. Carbon can make 4 covalent bonds. It can bond with each other to form single, double, or triple bonds with another carbon atom. It can also bond with other atoms such as hydrogen, phosphorus, nitrogen, and sulfur. It can form straight chains. It can form branch chains. And it can also form rings like those that you see in sugar such as glucose. Before we get into our biomolecules, you guys need to learn about our functional groups. Sometimes there are side chains that form alongside carbon backbones, and those are called functional groups. There are 3 essential functional groups that you guys need to learn about, and those are the ones that I gave you in the packet that I said make sure you know these. The first one is the hydroxyl group, which is OH. The second one is the carboxylic acid group, COOH. And the third one is the amine group, NH2. So as you can see up here, we have our hydroxyl group, the OH. We have a carbonyl group. This is a C, or a carbon double bonded to an oxygen. We have a carboxyl group, which is COOH. So here you see a C, O, O, and H. It's not always going to be nice and neat and in a single little nice and neat line. We're always going to have little branches that go out. So it's not just going to be C, O, O, and then an H. You're going to have some type of structure that you have to work with. We have our amino group right here, NH2, and then we have a sulfhydryl group. The S, sul, is standing for the S, and then hydryl is talking about a hydrogen. So sulfhydryl. We have several types of carbon bonds. We have our single bond, we have a double bond, and then carbons can also have a triple bond, as you can see right here. There are 2 ways of representing the different elements that you find in some type of compound. We have structural versus empirical formulas. So right here would be a structural formula. You see the actual structure of it, kind of like a building, or you could break the building down and see all of those beams. That's a structural formula. Or we can use an empirical formula. An empirical formula is just going to tell you the number of atoms that are represented by each. So for example, right here, we have C6H12O6. And if anybody knows what this is, you can shout it out to yourself and feel really cool. It's glucose. Alright, so this means that we have 6 carbons, we have 12 hydrogens, and we have 6 oxygens. We also have things that are called isomers. So some carbon compounds share the same empirical formula, meaning they have the same amount of carbons and hydrogens and oxygens, but they differ in their structural formula. For example, glucose and fructose are both represented by the equation C6H12O6. However, they have different areas where the atoms are located. Fructose is a sugar that's found in plants, just FYI. Polymers are large molecules consisting of many identical or similar subunits called monomers. So mono means one. Poly means many. Monomers, like I just got through saying, are single molecules. Polymers are formed by condensation of subunits with the removal of water, and this is called dehydration synthesis. So depending on the test that you're taking, they might use the word dehydration synthesis, meaning we are going to take water out of something. We're going to dehydrate it. When we take water out, things tend to stick together. The opposite of that is called hydrolysis. So for example, in a condensation or dehydration synthesis reaction, I'm going to take water out of something. So I just bonded something together. Now if I wanted to split it apart, I would add water. If I add water to that, it's going to split apart, and that's called hydrolysis. Hydro meaning water, and lysis meaning to split. Large carbon molecules are called macromolecules. That prefix macro means really, really big, like my eyeballs right now. And micro means itty bitty. So dehydration synthesis. If you look at these two monosaccharides right here, you see that over here we have an H being removed, and on this side we have an OH, or a hydroxyl group. The H from over here and the OH from over here combine to form water. So water is being removed from these two sugars. Once the water is removed, we form a bond right here, which links these two sugars together. When two sugars are linked together, we call it a disaccharide. Di means two, and saccharide means sugar. Like I was saying in the other slide, hydrolysis means splitting with water. So if we wanted to break that molecule apart, we need to add water. So polymers can be broken down by the addition of water, and we call that hydrolysis. Again, hydro means water, and lysis means to split. So if you look right here, we have sucrose, it is joined together, and this is a disaccharide. If we added water to it, we would get two separate molecules. And that's what we would use water or the process of hydrolysis to do when we want to split something apart. There are four main macromolecules that I'm going to be talking about. Those are carbohydrates, lipids, proteins, and nucleic acids. So carbohydrates are made out of carbon, hydrogen, and oxygen, and they have a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. They have a subunit called a monomer or a monosaccharide. Again, mono means one, and saccharide means sugar. So for example, glucose is an example of a monosaccharide, classified by the number of simple sugars. Sugars usually end in ose. So anytime you see that suffix ose, O-S-E, we're referring to a sugar. If you hear the word ACE, A-S-E, that indicates that there is an enzyme. So enzymes end in ACE, and sugars end in ose. So there are many different types of carbohydrates. We have monosaccharides, again meaning one sugar. Examples of monosaccharides are glucose, fructose, and these are major cell nutrients, and they're produced through the process of photosynthesis. So structures of common monosaccharides, we have glucose right here, we have galactose, and we have fructose. Moving on to disaccharides, di means two, and saccharides means what? Sugars. So two sugars. For example, sucrose transports sugars formed by the condensation of two monosaccharides. An example of that would be the table sugar that we eat. So all those sugar packets you put in cereal or coffee or cupcakes, those are disaccharide sugars. These are some common disaccharides. We have sucrose, lactose. If this sounds familiar, it should, because lactose is the sugar that's found in milk. We have maltose, we have isomaltose, and we have cellulobiose. Polysaccharides means many sugars. The prefix poly means many, and again, saccharide means sugar. In plants, starch is one of the most important polysaccharides there is. Starch in plants is where all of the nutrients are stored, or the food of the plant. The second type of very important polysaccharide found in plants is called cellulose. Cellulose is what composes the cell walls of plants. So think of cellulose, kind of sounds like something that you're going to find in a cell. So the cellulose is actually responsible for the structure of plants. Glycogen is a very important polysaccharide for us, for animals. So polysaccharides that we store in our bodies are called glycogen. And one thing I want to make sure you guys keep in mind is that polysaccharides in humans, the glycogen, is actually our short-term energy storage. So the role of carbohydrates in people, like I was just saying, the primary source is for our energy storage, and that's short-term energy. So if I was to go for a really quick run, that's what I'm going to be using up first, are all of my carbohydrates. After that, I'm going to dip into my fat reserves, and I'll be using and burning up my fat, or lipids. Like I got through saying in the previous slide, it's used as a structural component in the cell walls of plants. The next biomolecule we're going to discuss are lipids. Lipids are made out of carbons, hydrogens, and oxygens. However, there are very few oxygens. They are insoluble in water, meaning they're not going to dissolve. So if you remember the water lab that we had, when we added oil in the water, the oil always floated on the top of the water because it is non-polar, meaning it doesn't have a charge. Whereas water, which is polar, it has a charge on the oxygen that's slightly negative and a charge on the hydrogen that's slightly positive. So they form many carbon-hydrogen bonds, and we call these hydrocarbon tails. One subunit is called a glycerol, and we have 3 fatty acid chains. So you may have heard the term triglycerides if someone in your family has trouble with weight or with blood sugar. A lot of times they're going to have trouble with fat in their bloodstream, and they'll go get a blood test and say, oh, you have high triglycerides. So tri means 3, and glycerides is just referring to those fatty acid chains. So when I say triglyceride, this is exactly what I'm talking about. We have our glycerides right here, and there are 3 of them, these 3 fatty acid chains. Now the fatty acid chains are going to be hydrocarbon chains with hydrogens and oxygens. The role of lipids. So lipids are there to store energy. Energy is the long-term storage for lipids, and it has 3 times the amount of energy that carbohydrates do. Lipids are found in membranes. That's why we call it the phospholipid bilayer, because it's made out of 2 layers of fat. It helps in insulation. So if you see marine mammals, a lot of times they look a little bit chunky. There's a reason for that, because they need that layer of fat or blubber to protect them from the cold water. So it's an insulator. It also insulates our organs and helps protect them from trauma. There are 2 forms of fatty acids. Saturated fats. These do not contain any double bonds, and they are completely solid at room temperature. So for example, butter and then lard. You see the lard in those big buckets, they scrape it out, it's white, really nasty. That's what they use to fry a lot of your foods in. Not very good for you. And then we have unsaturated fats. These are the good fats that you want to try and incorporate into your diet. Unsaturated fats have double bonds between carbon atoms, and they are liquid at room temperature. So for example, vegetable oil would be an unsaturated fat. So if you look here, we have saturated. meaning that all of the carbons have single bonds attached to them, either to another carbon or a hydrogen. Unsaturated, if you see right here, they're not fully saturated with those hydrogens, so they're a little bit lighter. So think of saturated as you're really, really heavy and they're going to weigh you down. Unsaturated is going to be light, and those are the healthy fats. The next biomolecule we're going to discuss are proteins. Proteins are made up of long chains of monomers called amino acids. There are 20 common amino acids, but several combinations can build thousands of proteins. Kind of like we only have 26 letters in the alphabet, but out of those 26 letters, we can form hundreds of thousands of different words. The role of proteins are that they are the building blocks of most structural components. One very important thing I want you to make sure you know about proteins is that enzymes are proteins. And enzymes are what catalyze reactions or what speed those reactions up. An example of a protein would be meats. So the way you can tell if you're looking at a protein are you're going to see this amino group right here, that NH2. Remember when we were talking about functional groups at the beginning of this presentation? Well, this is how they apply. This is our amino group, our NH2. It also has a carboxylic acid group, that COOH, and you see that right here. We have our COOH. So the types of proteins, a peptide is one amino acid. Two amino acids linked together is called a dipeptide, and many amino acids linked together, as you may have guessed, are called polypeptides. The last type of biomolecule we're going to discuss are nucleic acids. These make up long chains of monomers called nucleotides. Nucleotides are composed of a phosphorus or phosphate, a 5-carbon sugar, and a nitrogenous base. There are two types of nucleic acids. There are DNA and RNA. DNA is called deoxyribonucleic acid. It is double-stranded and it stores all of the genetic information of an organism. That's what tells us, are you going to have green eyes or blue eyes, brown hair or blonde hair? You're going to have a big nose or you're going to have a small nose? Are you going to have any type of diseases or genetic disorders? That's all in your DNA. RNA is a ribonucleic acid. It is single-stranded, and it has a big role to play in protein synthesis. So RNA and DNA. Right here we have DNA, which is double-stranded, as you can see right here, and RNA is single-stranded. Another difference between RNA and DNA is that DNA has the nucleotide bases of adenine, thymine, guanine, and cytosine, while ribonucleic acid, or RNA, has adenine, guanine, cytosine, and it has uracil instead of the thymine. So both DNA and RNA are made up of nucleotides. DNA is double-stranded, while RNA is single-stranded. The nitrogenous bases in DNA, like I just got through saying, are guanine, thymine, cytosine, and adenine. In RNA, thymine is replaced by uracil. So digestion. What happens to these biomolecules whenever they enter our mouth, like when we eat a Happy Meal? So we eat the food, we take it in the form of carbohydrates, proteins, and lipids. With the help of special proteins called, what? It ends in ACE, enzymes. We break down these macromolecules into monomers. These monomers are absorbed into the bloodstream in the small intestine and are then carried to the cells where they can be used for energy. This concludes our slideshow. I hope you enjoyed it. This is the Queen Nerdling signing out.